So, good morning, ladies and gentlemen. I'm sorry, Mr. Chairman. So, first of all, let me express my gratitude to Khalifa International Award for uh, organizing this beautiful conference and inviting me. Thank you very much. So, I would like to tell you about our job, about our innovation technologies for date palm protection against red palm weevil. So we know that FAO
Excuse me, please.
estimate global production losses due
Doctor,
to
please. Excuse me, please.
yes.
Would you please introduce yourself?
Yes, definitely.
Thank you.
So my name is Edward Krzyżanowski. I am doctor professor in St. Petersburg University, and we also making a lot of devices in agriculture, in medicine, and today I would like to tell you about our innovation in agriculture. So thank you very much. So our company name is Palmolife. It's from St. Petersburg, Russia. So FAO estimate global production losses due to diseases up to 30%. So RPV is the most destructive palm pest, and it spreads very, very fast. So conventional infestation is quite limited due to specific habit of RPV. And of course, it became big problem. So we have our solution. It's two devices and one platform. So first device is smart trap. One can identify the injection foci, and the second one is palm guard device, which can protect palm using specific electromagnetic field. It is scientifically proven. And definitely we use AI and IoT system. It's one platform, and together it consists one closed network. So first of all, I would like to tell you about our smart trap. We developed a specialized sensor that count falling into the trap pests. And this information directly comes through our software, remotely to the person who controls these processes. One have solar battery, so we can live very, very long time, and Wi-Fi module, which is communicated with a server. So another device is standalone device, which is placed on the trunk of a palm, and it provides electrostatic protective barrier, which is scientifically proven. This device also have acoustic monitoring features. It's listening what is in the trunk and what is in surround area to detect potential infestation earlier. So we developed smart trap software. All information about the number of pests that fall into the trap, it's analyzing by AI and can be monitored remotely. So also it's available on Play Market under OS, under Android, so we can use it. So the system smart traps allows you to monitor an arbitrarily large area. So the technique looks like the next. So first of all, a smart trap system detects foci, detects the area which are under the risk, and then we put on in the area of risk a special device which name is Palm Guard to protect this area. We have intellectual property in Russia, we have application in UAE, Saudi Arabia, Qatar, Iran, and also we have publication in Blessed Tree Journal. It's a publication article about our study in IGBA. So we did study during 1 year. So I'd like to tell you about it a little bit. So we put our devices in 3 zones. So first zone was low infestation zone. Second one was medium infestation, and third one has high infestation. So monitoring dynamics of RPV appearance was conducted using Smart Trap, our sophisticated software, Palm Guard acoustic monitoring, and also visual inspection and continuous audio and video recording. So afterwards we analyze it everything. In low infestation area, we didn't detect any RPV activity during 11 months. In medium infestation, RPV activity detected in 3 trees. But protection function, after we install the protection devices, after activation, the result was the next. The RPV activity fully eliminated in 2 months. About zone 3 in high infestation, we compare definitely areas with our devices and without our devices. So no device and no device we see on the graph. So after 11 months of monitoring, we see that data, we see this graph, that RPV activity was drastically eliminated, drastically decreased in 2-3 months. It was significantly smaller than previous one. But in no device, it was not any positive dynamic. So we can conclude that electromagnetic barrier works very good and effective, and it drastically decreases RPV activity. So now we are working on our next generation traps. It's integrated, so it's prototype, it's integrated with drone. We have some video, but not working here. So anyway, we integrated with a drone. So we put it more powerful sensor with artificial intelligence. One can record photo and video. Now it's using ZigBee, not Wi-Fi, so it's a more economic device. It has built-in GPS. Now they can navigate autonomously to infestation zone by ourselves using AI. They be able to self-organize for optimal protection using AI. So it's self-organizing system. So they can fly to the Weevil collection point, unload this trunk in special container, and then process them. So now it works. You see prototype. So it's new generation smart trap. So we would like definitely to open production facility in UAE with collaboration of Khalifa International Award. And of course, we have some success, and definitely we would like to integrate with UAE government for environmental protection programs. And definitely would like to cooperate with the largest plantation in UAE and all MENA region. So our team contains 7 doctors in mathematics, in physics, in biology, many engineers and programmers. And I would like to also express my gratitude to our friends, His Highness Sheikh Al Hassan Al Noaimi, because he was the first 5 years ago. He told us about this problem with RPV. We started to make our first approximation devices, and now we have statistically significant results, and we are very happy about it. So, thank you very much. Thank you.
Thank you. Thank you, Dr. Edward. This is really very important, and this is a very good innovation. For the sake of time, we will take the questions by the end of the session. So please keep your question until the end. And thank you so much.
Thank you. Thank you so
Dr.
much.
Abdullah.
Yes, thank you very much, Doctor. Our second speaker, he will be making his presentation online, and it's Mr. Dr. Ali Zaki Abdul Qadir. Is he ready? Dr. Zaki Abdul Qadir, he will present first report and biological characterized on I'm sorry for this, teratobditis, okay, which is an as entomopathogenic nematode against the red palm weevil on date palm in Iraqi region. Is he ready, please? Okay. Okay. Okay, then we will go to the next one. Mr. Omar Hisham Rida Khidr. أنا مانيش حظ مع الجماعة هذا. كل واحد يتكلم في اللي بعده. Okay, his answer. Yeah. Mr. Tushar. Mr. Tushar. Mr. Tushar Chaudhary. Yes, yeah, okay, good. Yeah, please introduce yourself before starting your presentation.
Good morning, everyone. Thank you for joining us today. My name is Tushar Chaudhary. I'm from IIT Ropar. I'm a final year BTech student in chemical engineering. I'm presenting here on behalf of our team AgriScience based in New Delhi, India. Our presentation titled An Adaptive Acoustic Surveillance System for Red Palm Weevil using a hybrid AI approach. To give you a sense of scale of the problem, India has nearly 60,000 acres under date palm cultivation, which has an infestation rate of 6 to 8% reported across several plantations. The encouraging part is that our hybrid AI system achieves 89.1% accuracy while reducing false alarm significantly from 15% down to 4.8%. Over the next 15 minutes, I'll walk you through how we achieved this from field data collection to model deployment and finally our results. Here, how I'll structure today's talk. We'll begin with the background, why date palm sector faces unique challenge with RPW and why detection in real field condition is particularly difficult. Next, I'll walk you through with our data collection processes across 3 different agroclimatic zones in India. I'll also explain how we processed acoustic data and converts them into format of AI model that can interpret. From there, I'll cover how we built and refined our model and why we moved beyond a standard deep learning approach to a more reliable hybrid approach. Finally, I'll share our key results and outline with our next steps ahead. Let me first set the context for why this work truly matters. The red palm weevil is a major threat to palm cultivation. Infestation rates of around 5 to 8% have already been reported across multiple sites. What makes it particularly dangerous is that larvae feed inside the tree trunk, completely hidden from the view. By the time visible symptoms appear, the damage is often irreversible. We don't know that we do know that red palm weevil generate distinctly acoustic signals as they feed. However, in real field condition, these signals are heavily masked with environmental noises, wind, rain, machinery, and other biological sounds. There are significant another challenges. Most existing detection systems have been developed for regions like Middle East or other Southeast Asia. But India's agroclimatic conditions are very different, and until now, there has been no validated field ready solution tailored for Indian farms. This is a gap we set out for address. To address this challenge, we developed a device called Strider. It is a portable handheld device designed for untrained farm workers which require no specialized skills. This device is completely non-invasive. A probe is simply placed on the surface of the trunk, no drilling, no damage to the tree. What makes Strider especially powerful is at its multimodal capacity. It can capture acoustic signals along with some complementary metadata, simultaneously enabling more reliable detection. And importantly, the system has been field validated across different detection agroclimatic zones in India, demonstrating robustness in device real world conditions. The data behind the system was not collected in lab, it was gathered in real field condition over more than 2 years from May 2024 till now. Our team collected over 4,500 recordings across different agroclimatic zones: Rajasthan northwest, Goa in west coast, and Tamil Nadu in south. These regions differ significantly in humidity, temperature, and ambient noise. This diversity was intentional. We wanted to ensure that the model is robust and performs reliably on varying real world conditions. Importantly, all recordings were collected by untrained laborers, workers, not the scientists. This is a deliberate choice because any scalable solution must work on the people who are actually present on the farms. Along the way, we iterated on the both hardware and data. We developed our first device early in 2024 and began initial data collection. Based on learning of the regions like Tamil Nadu and Goa, we refined our model system and the second and third version, improving both data quality and model performance with each iterations. Yeah, I can show you the video that labor is collecting the data. This is the device Strider, and he's the only one who is collecting the data from the tree. We have a specific tree ID so that we can tag the geological data for it as well. This slide completely explains the two-step process at our core our system it is. The step one, we convert our raw acoustic signals into visual format that the AI can understand. We use MEL spectrograms which transform sound into an image which has a frequency pattern becomes clearly visible. Step two, we pass the representation through a two-layer classification pipeline. The first layer is the convolutional neural network, which analyzes the pattern and predict whether it corresponded to the red palm weevil activity or not. The second layer is a physics-based dynamic frequency gate domain. This addresses a critical challenge. Wind noise are often mimic larva feeding signals and can mislead the model. By introducing a deterministic frequency domain filter, we effectively separate true RPW signal from environmental noise. The outcome, the false positive reduced significantly from 15.1% to 4.8%. This slide compares our model performance when trained on two different types of data. On the left, you can see that we are using synthetic lab generated data, and on the right you can see the results from our own data that we collected from our fields. The difference is clear. Performance improves significantly when the model is trained on our real data field collected over the actual Indian conditions, rather than synthetic data from the environment conditions. This highlights a key insight: context matters. A model trained on synthetic data or region-specific data does not generalize well to real-life conditions in different environments. That is precisely why our 2 years of on-ground data collection has been so critical. It enables reliable development and deployment-ready performance in the field. Here are the results comparing our 4 models approaches we evaluated. We begin with BGG16 as a baseline, a well-established deep learning architecture. However, we achieved only 54.3% accuracy along with high false positive, making it unsuitable for field deployment. ResNet showed us improvement, reaching 69.9% accuracy, but still suffered from a high number of false positive. A custom CNN trained directly on field data performed significantly better, achieving 83.6% accuracy with a false positive rate of 15.1%. Finally, our hybrid approach with a frequency gate delivered our best performance: 89.1% accuracy, 84.2% precision, and we are also extending our platform to detect other tree boring insects, moving beyond RPW to create a more comprehensive plant detection monitoring system. From a commercial perspective, we aim to begin sales and both services deployment in second half of 2026 across India and other GCC markets. Thank you so much for your time and attention. To summarize this, we have developed a field validation non-invasive acoustic AI system for early detection of red palm weevil in date palms. Our hybrid CNN model achieves accuracy of 89.1% with a low false positive rate of 4.8% and has been tested across different regions in the India. We believe this technology is highly relevant to the GCC and UAE context, and we are actively seeking partners and collaborators for field validation in the region. If you are interested in learning more and exploring potential collaboration opportunities, please feel free to reach us at info@agriscience.in. I'm happy to take questions, and my colleague Joshua Kaj is also here to assist you.
Thank you very much, Mr. Chaudhary. Thank you for presenting this promising technology. I'm sure that it will raise questions also.
Yeah.
And the discussion, as the doctor said, that it will be at the end of the session. Thank you very much.
Thank you.
Well, thank you. Is Dr. Ibrahim Arafat here? Okay. Well, our next speaker is Dr. Ibrahim Arafat. Please, if you introduce yourself, and you have 15 minutes. Thank you.
Good morning. I am Ibrahim Arafat. A senior researcher from the Agricultural Research Center, Egypt, PhD from University of Cordoba, Spain. First, I would like to thank the organizing committee and the United Arab Emirates for their strong support of scientific research for this conference. Today I present my work. I present my work, our study about the rosmarinic acid as potent natural biopesticide agents, red palm weevil. This work was conducted in collaboration with two colleges, Dr. Ahmad Dabour and Dr. Mona Hassanin. The red palm weevil is a major challenge for the global date palm sector is a destructive base affecting over 40 palm species across more than 49 countries, causing losses about $2 billion annually, with increasing resistance to a chemical insecticide. Rosmarinic acid is a natural compound found in many plants, was investigated as a safer alternative using HPLC analysis. In the first, we quantified its content in 6 plants, ornamental plants, from family Raceae. For this bioassay, for this analysis, the oregano and the lemon palm showed the highest concentration, 46.7 milligram per gram draw weight, highlighting their potential for large-scale extraction. For the laboratory bioassay, rosmarinic acid was tested at 3 concentration, 3, 4, and 5 milliliters per liter water, and was used the deltamethrin as a reference or control. The result of this bioassay, first mortality was recorded daily over 10 days. Results show the time dependent increases in mortality, indicating a cumulative toxic effect. The force nestor larve was the most. This is in this figure. The force nestor larve was the most sensitive stage to the rosmarinic acid, while the adult, in this figure, the adult was the more tolerant to the rosmarinic acid. At 5 milliliter concentration of rosmarinic acid, achieved mortality comparably to our extended digital screen in this figure as cumulative mortality. In the toxicity analysis, mortality data were corrected using Abbott's formula and analysis by probability analysis. And the LC, low concentration, 15 value was 2.004 milliliter per liter water for the force interstellar, confirming high sensitivity and the statistical analysis significance was validated using Pearson chi-square test. Regarding the mode of action of rosmarinic acid, the second biocide, we used force and XR and stellar were alimented with rosmarinic acid at LC 50 concentration. After during 72 hours post feeding natural indicid. We in this bioassay, the biochemical assay is infested. The larvae were treated with this concentration, and we showed in this we showed the inhibition of gross indices of this larva in the for force in stellar, confirming the high sensitivity statistical and reduce the larvae feeding and growth in this parasite with especially with the larvae with the force in stellar. And in the other parasite, we study the physiological impact of rosmarinic acid at LC 50 concentration.
Dr. Ibrahim, 2 more minutes, please. دقيقتين كمان.
On host detoxification defense mechanism. In this bioassay, we as a result we show that the inhibition of detoxification enzymes, especially with the force in stellar. Finally, we studied the effect of rosmarinic acid in the field. We treated the palm with injection of the rosmarinic acid in the gallery of insect. In this experiment, we can see in this table the high percent of recovery of palms after 30 days, recovery rates reached up to 83%, while at 5 concentration of rosmarinic acid. This finding demonstrates that rosmarinic acid is a promising eco-friendly and effective biopesticide for controlling the palm weevil. Thank you.
Okay. Okay. Well, thank you, Dr. Ibrahim, and we will be taking questions in the session. So thank you so much. Is Dr. Bino here? Dr. Bino Antoni. Is he coming? Please. Okay, Dr. Bino, please, the floor is yours, and you will be having 15 minutes, please. And please introduce yourself at the beginning of your presentation.
Hi, good morning all. I'm Dr. Bino Antoni from Khalifa Center for Genetic Engineering and Biotechnology, UAE University. So this presentation covers my past research at King Saudi University, where I was working. I just recently moved to Khalifa Center. So the title of my presentation is Olfactory Biosensors for Early Detection of Infants, Palm Weevil. So, as you all know, we are discussing lots about the palm weevil. So, palm weevil is distributed all over the world. As you can see that, especially in North Africa, Middle East, some part of Asian countries. So, here you can see the recent review article published from our group. So, the native range, as you can see, the pink color. And the invaded range, as you can see, the red color. So it is almost distributed all like Middle East and North Africa. So the symptoms, as you all know, there is presence of grubs and larvae at the base of the frond, the typical tunneling, and also finally the tree will be dried. So, we started a research on developing olfactory concept. So usually when there is a stress, palm trees are there, so they emit the volatiles. So there are so many volatile organic compounds. So same like in the infested palm trees, there are the colony built up inside. The weevil, the male weevil, they produce the pheromones. The two compounds like ferruginol and ferruginones. So this, we call it like volatile organic compounds. This is what is called palm volatilomes that contains the pheromone as well as the stress palm emitted volatiles. So early detection is very important to save the tree. So why don't we track these volatiles? So when there is infested trees are there in the field. So why don't we track the infested trees like tracking these palm volatilomes, like the pheromones and the chiromones, the palm volatilomes. So using like the sniffing device. So for that we need to have a sensor or a sniffing device. So detecting the volatile releasing from the infested trees. So that we know which tree is infested among hundreds and hundreds of the trees. So here comes the importance of developing the biosensors. So for that we need to unravel the detection, pheromone or pheromone detection mechanism in palm weevil. So palm weevil, the nose is antenna. So antenna has a small hair-like structures called sensilla. So each sensilla has a nerve innervations. There are two important proteins. That is, one is odorant receptors and the co-receptors. Then the odorant binding proteins. The odorant binding proteins carry the pheromone or any ligand molecules towards the receptors. So when it binds to the receptor, signal will be transmitted. There is a depolarization, repolarizations. So the odorant receptor and co-receptors works like a ligand-gated ion channels. So when there is a ligand interaction, so the gate will be open, as you can see here. The gate will be open, and the influx of the calcium ions, and so the depolarization will happen. So we characterize, for the past many years of the research, we characterize the odorant binding proteins, odorant receptors, odorant co-receptors, so all of them are published, and the pheromone receptors and pheromone receptor also. The pheromone receptor is referor-1, that is the OR1, odorant receptor 1, and the odorant binding proteins that is tuned to the different the pheromones and the pheromones. So the first concept is, suppose if you can take these pheromone receptors and co-receptors, if you transgenically express this on cell systems, cell-based sensors we can develop. So we use the HEX cells, human embryonic kidney cells, so fabricated with the transgenically expressing the pheromone receptors and pheromone co-receptors. The same method, these cells genetically encoded with there is a GCAM proteins, so that when there is a calcium influx, it will produce the green colors. So that we know the pheromone presence or pheromone presence it is there. So here it is for detecting the pheromones. So that is what it is explained here. So these cells were immobilized in a micro array plates with hydrogel matrix. So as you can see that when there is a liquid phase pheromones or the vapor phase pheromones, if you supply the liquid and vapor phase pheromones, so it is detecting the pheromones. So this paper we published in Biosensors and Bioelectronics just recently. It appears in last year. This year, January. And so that we can detect the pheromones and pheromone presence in the field. So this is in collaboration with Tokyo University, so funded by Ministry of Education, Saudi Arabia. So second concept is illustrated here. As you can see that the infested farm and healthy palms. So infested palms, usually there are colony of the weevil. They emit like the pheromones, and the stressed palms also emits the pheromones. So there are devices like QCM, quartz crystal microbalance, where we can immobilize the pheromone receptors and pheromone receptors or the pheromone binding proteins. So when there is a molecular interactions, the frequency shift we can detect using the machine learning techniques, so that we can identify the healthy and infested palms. So the pheromone receptors and pheromone receptors, as you can see that this is this is immobilized on QCM device, and so finally by using the IoT device, so we can transfer this the information to a mobile device, and so real-time information we will get it, real-time detection we will get it. So we synthesize the odorant binding proteins. There are two odorant binding proteins that was involved in pheromone detection, and there is ethyl acetate detection. So the OBP 1768 and OBP 23. And we also, like pheromone receptors, we synthesize the peptides. So we mobilized on the QCM device, so that this odorant binding proteins and odorant receptor will be stable for like 7 months or something. So that will just works like a insect nose. So it can detect the pheromones and the ethyl acetate emitted from the stress palm trees. So we conducted a field trial, the first field trial, and so we detected we collected the volatiles in and around from the infested palm trees, and these volatiles, usually the detection, the peaks we will be getting like this, and so when there is the sensor can be detect up to the level of 60 ppb parts per billion level. Orders they can detect, and so we finally made a sensor array that is fabricated with the odorant receptors and odorant binding proteins. There are two kinds of odorant receptors we used based on the nanodisk integrations, and also odorant binding with mutant and wild type odorant binding proteins we used. So we are getting the real time detection of the infested palm trees. So here is the result. As you can see that when you compare with the uninfested trees, there is a clear differentiation. So box plot here it is showing the sensor response to the infested and non-infested trees. So this project was funded by KAUST in collaboration with the different universities in Manchester, INRE Paris, University of Rome, and so this is the portable sensors. So this patent is under considerations and publication is under review. And so I want to conclude my presentation. Biosensor array is capable of detecting and discriminating the red palm weevil pheromones. Sensor cells, chip-based sensor cells that can detect the vapor phase as well as the liquid phase pheromone compounds. So sensor cell array, the vapor phase at level of 0.1 to 100 ppm level we can detect. And the biohybrid sensors, the sensor array, the last one, the QCM based, as I mentioned, it can detect 60 ppb level pheromones and the ethyl acetates. So practical application of this sensor is early detection of palm weevil in the field. So to conclude my presentation, I would like to thank all my students and collaborators and finally the fund agencies. Thank you all.
Thank you, Dr. Bino. Actually, we will keep the questions until the end of the session, so we can move ahead with the time. Dr. Abdullah.
Yep. Our next speaker is Mr. Dr. Waqas Wakil. Is he here? Dr. Wakil. Okay, the next one is Muhammad Abdul Rahman. Muhammad Abdullah. Rahman Gol. Dr. Rahman Gol. Yeah. Please introduce yourself first, please.
Good morning. I am Dr. Weevil Rahman. My research focusing on the intersection of entomology and agricultural science, specifically looking at the host-based biological response of red palm weevil and selected dead palm varieties. Today I am excited to explore my finding on how different dead palm varieties influence the behavior and development of this pest. This slide shows the introduction of date palm. The date palm Phoenix dictyria genus belongs to palm family Ericaceae, is an important fruit crop among the globe. Pakistan is the fifth largest producer and third largest exporter after Iran and United Arab Emirates. Date palm have a secret and religious importance. The Holy Quran mentioned that date plant has no harmful effects being grown on its vicinities. This slide shows the symptoms caused by red palm weevil. It is very difficult to recognize the first sign of red palm weevil presence. Later, its stages of attack are conspicuous. However, damage done before these later stages, when detection becomes easier, can suffice for palms to die. A very typical sign of red palm weevil is distorted crown in case of severe damage, which can be easily seen as compared to the other symptoms of weevil infestation. This slide shows the following treatments, including asil, muzafti, gulistan, majul, and halavi. This study was conducted in the laboratory of entomology, Gumag University, Ismail Khan, including asil, muzafti, gulistan, majul, and hawali, which were used against to investigate the relative resistant susceptibility against red palm weevil. The soft portion of each variety were placed in transparent plastic jar, having size 4 cm long and 30 gram weighing. The weevils were reared in the jar, and the jar was labeled by the name of variety. The data were collected on egg hatching percentage, total larval duration, pupal duration, fecundity, and adult longevity. This particular slide shows the egg hatching percentage of red palm weevil when the weevils are reared in the respective host. The maximum egg hatching percentage was noted when the weevils were reared in the variety asil, having 88.7%, which was followed by gulistan. The minimum egg hatching percentage was noted in the variety halavi. This slide shows the instar duration from 1st to 7 instar. This slide shows the total larval duration, pupal duration, fecundity. The highest fecundity was noted in the variety gulistan, which was followed by muzafti. The minimum significant was noted in the variety asil, which was statistically different from all other tested treatments. The second line showing the larval duration. The highest duration was noted in the variety halavi, which was followed by gulistan. The minimum was noted in the variety asil. At the last one, the maximum it's not showing on the screen. Not showing on the screen. Thank you, Soria. This slide shows the survival percentage, fecundity, and weight loss of red palm weevil was structured in different hosts. The first green line showing the highest survival percentage was noted in the variety asil, which was followed by gulistan and majul. The second red line are showing the fecundity, when the weevils were here in the host of Assyria, which was regarded the maximum fecundity. The minimum fecundity was noted in the Harawi. In weight loss, the maximum weight loss was noted in the variety Asil, which was followed by Gulistan and Majul. The minimum weight loss was noted in the Harawi. This slide shows the lifespan of red palm weevil, male and female. The highest lifespan was noted in the variety Asil, having 103.14 days, which was followed by Gulistan. The minimum male adult longevity was noted. The female longevity was noted. The maximum 83.40, which was statistically different from all other tested treatments. Final reflection, the relative resistance of different cultures revealed that the Assyrian variety was found most susceptible for the development, survival, and reproduction of red palm weevil, whereas the Harawi variety had a negative effect on the biology and survival of tested insects. Thank you for your attention.
Thank you. Thank you, Dr. Ghulam.
Okay, is Dr. Zilal here? Zilal. Okay. Is the online working or it's not working? Okay. Okay, let us... Well, what do you think? We go for discussion or we continue with the other presentations? We continue, is it okay? So let us continue. Is Mr. Robert Flyers here? Mr. Robert. Okay. Dr. Khalid Masmoudi. Okay. Dr. Abdul Rauf Muhammad. Okay. Dr. Muhammad Kamal Abdul Latif. Dr. Muhammad Kamal Abbas. Okay, well then let us take the time now for discussion, because maybe the people, they just signed themselves for the afternoon session, so they will come to the afternoon after lunch. So let us open the floor for discussion. If somebody has a question or would like to make a comment. Okay. Most. Well, just to raise an issue. Since we are really going in different directions, from biological ways to artificial intelligence to all of those methods. I mean, and some people, they are in the private sector. To which extent really we see ourselves we are going in the right direction, and we will reach a situation where at least we achieve a lot of control of the red weevil, especially one of the problems is related to the management situation, because when you go to trees used in gardens, used in landscape, some of these trees, I mean, this is difficult to control because they are not in farms and people are not dealing with them as producers or something like that. So can somebody elaborate on this, please, from the speakers? Okay. Yes, please.
Thank you, sir. Thank you, sir. Respected sir, always the farmer are thinking that we have going to the way of economically. As the resistant variety shows that the red palm weevil has not been preferred to that site. If they are going in that site, but it will be lost very difficulty, because if you know the Gulistan variety has found more sugar contents, the red palm weevil has going to very quickly, but you have used on behalf of this variety Harawi, which was susceptible against red palm weevil. Thank you, sir.
Based on your results,
Yes, sir.
based on your results, can we talk about resistance here?
Resistance here.
Can we talk about resistance? Because when we see your results, the infestation rate, even for those who, the one you said it's not very sensitive, the rates are very high also.
Yes, sir. Respected sir, those variety having the low contents of sugar, the weevil will not attack at there. If the variety which are going Muzafti, Gulistan, Begum, Jangir, Daki, having more type of sugar contents, the weevil will attack directly on that side. If you have using the Harawi module having a low sugar contents, so that's why.
Okay, well, thank you. Does anybody have any comment or suggestion? Please. Can you be on the podium, please?
I am Dr. Atik Ahmad Alizai. I am from Gomal University, Pakistan. Mostly I work in date palm orchard management. Actually, I am horticulturist basically. But since we have always contact with the growers, and we see only this problem everywhere in our country as well. Lot of people are working on biotech, on AI system. Lot of things they want to identify before it damages the plant. But I think we have already traditional system as well. Many of our workers, skilled workers, our scientists, they can detect very easily when they go into the orchard, and we can detect that okay, these plants are just close to infestation, and they will get infestation very soon. But the problem actually lies with our management system, with the farmers, because we mostly do not regularly monitor our orchards. If we have AI system, if we have traditional system, if we have any other biotech system, if we do not do it regularly, I usually suggest my farmers that we should at least keep record 3 or 4 times a year, go to the orchard and monitor the orchard, look to the our plants, and we will easily detect that is our plants are infested or not. So if we can manage to have our farmers keep your monitoring record with them, I am sure that they will get rid of this problem very easily. So this is
From your experience, I mean, and different countries, there is really a breakthrough in the control of red weevil, or still, I mean, the problem is it steady, is it increasing, is it decreasing at the level of countries, and this is the situation.
yes, see, we are controlling, and we are using different techniques. I think every week, every other month. Just few days ago, we treated orchard in our areas. I would say two of the orchard I treated with few pesticides. But because of these non-monitoring systems, so it is increasing day by day. If we monitor the plants and regularly treat our trees without having thoughts of if this orchard is infested or not, we should go and monitor and treat our trees regularly as we produce and provide them the fertilizer. So easily it could be managed, I think. So this was our opinion. Yeah. Thank you.
خالد المصمودي، نعم دكتور خالد المصمودي، نعم. دكتور خالد, please introduce yourself. Thank you.
Good morning, everyone. Do you hear me? I'm Khalid Masmoudi. I'm a professor of plant biotechnology and plant microbiology. I'm the vice dean of the College of Agriculture and Biomedicine at UAU. Today, first of all, I would like to thank the organizer of this 8th meeting on conference on date palm, International Date Palm Conference in Abu Dhabi here, UAE. My talk will be about the development of a novel heat-stable biopesticide. Formulation by utilizing date palm-derived layer-2 dehydrogen proteins for sustainable management of the devastating red palm beetle. This work was done with at UAU, and I have with me my former PhD student, that now he's pursuing as a postdoc. Just a very brief introduction about red palm beetle you are very familiar with. I'm not entomologist, I'm a biotechnologist, and it seems that red palm beetle is causing a lot of damage to the date palm, and it's spreading very fast from farm to farm, and the larvae goes into the trunks and causing internal destruction and date palm mortality at the end. the current pest management control used actually is a broad spectrum of chemical insecticide to prevent and mitigate the infestation. This causes a problem to the environment and to the farmer pocket money. At the ecological concern, there is a hazardous of both ecosystem and public health. There is a limited success in biological control for the red palm weevil, like using the entomopathogenic fungi like Bovaria bassiana. It was a very big hope, but still not giving successful. There is also nematodes and also many others biological controls tested in the farm. Came the BT, the Bacillus thuringiensis based control, which produces the insecticidal delta toxin and is a promising, environmentally friendly, and for sustainable biological control agent to control the red palm weevil. However, as you know, the BT is not stable. It gets damaged, gets destroyed immediately when you spray. So people try to spray the BT for the hot country, even late in the afternoon, early evening, or early in the morning to avoid the destruction of the BT. Also, it's very sensitive to UV light. That's why you cannot see BT used in UAE or in GCC country. The mechanism of action of BT is when the insect ingests the BT, the toxin gets activated by proteases from the gut of the insect, and the cry genes, which are the most effective, will be activated and destroy the mid-gut cell lining and causing the stopping of the feeding of the insect, of the larvae, and at the end they will die. Larvae and adults also. Effective strains for BT to control the red palm weevil is the most important is BT tenebronosis subspecies. Specifically toxin like Cry1A, which have been shown high toxicity to the red palm weevil larvae. We were working in my team before joining UAE for a long time on dihydrin and Rea2 proteins. And we isolated the first dihydrin from wheat, and we found that this dihydrin has no structure, it's a disorganized protein, and when we tested in some enzymes, it brought some heat resistance, heat thermostability to the enzymes. So dihydrin are stress-responsive proteins in plants. They act as a molecular chaperone to prevent other molecules. They have no structure. They took the structure of the interacting proteins, and they prevent the aggregation and the heat stress. Dihydrin are a member of a large family of Lea proteins. I would say Lea2. And they accumulate in desiccated seeds or vegetative tissue challenged with abiotic stresses. They are involved in water osmotic stress tolerance, in salinity tolerance, and in heat stress. The molecular function of this dihydrin is still unknown, but we have some clue. As you can see, dihydrin constitutes a distinct group, biochemical group of Lea2, and they are characterized by some signatures, like the YSK segment, the tyrosine, the serine, and the lysine track for the characterization of this dihydrin. And we have either a YSK segment, YK segment, K segment only, or SK segments. These proteins are highly phosphorylated. It goes from the nucleus to the cytoplasm. Then it makes the shuttle back get phosphorylated go back to the nucleus and get dephosphorylated, of course. When we analyzed the date palm genome, we found that this layer 2, this dehydrin, as you can see, the blue color is layer 2. The others are the other layer. These are highly predominant in the date palm genome. And compared to other crop genome, layer 2 from date palm are highly expressed. We have 62 members in date palm compared to rice, which the genome is sequence 2, with a 50 or 52 members. Here we, just to give you an update about dehydrin isolated from wheat, DHN5. It was expressed in PGX vector, overexpressed, purified, and used in enzymatic cocktail to test the BGLG, the beta-glucosidase, which is a very important enzyme at the industrial level. And as you can see, I don't know if there is pointer. When you use the BGLG and you treat it at 70 degrees, the reaction, the enzyme gets degraded after 20 minutes, almost. But when you add the DHN5 protein that you overproduced, it gets the enzyme will be, of course, degraded, but later on. So there is a gain, there is a win of time of the enzyme. That's why you don't have to add the enzyme often to the reaction. And this was tested in different other enzymes like LDH, like PUD, and so on. So the objective from this study was to use this dehydrin isolated from date palm to, as a complement to the biopesticide formulation that is present in the market. So we use the layer 2 to protect the Bt toxin from the thermal degradation, evaluate the layer 2 mediated enhancement of Bt, and to measure the insecticide efficacy against red palm beetle eggs, larvae, and adults under normal and heat stress conditions. The proteins were cloned, overexpressed in PET vector, and purified using histidine tag, and was used to monitor the red palm beetle mortality. So here we have some. We were catching the red palm beetle, either larvae or adult from Silel in a line in El Foix, and we tested using the Novodor biopesticide, and we added the layer 2 protein, the dehydrin, at a ratio of 1 to 40, and we tested in a leaf dipping on date palm fonts. This is the control was done at 25 degrees, while the heat stress was done at 42 degrees. Of course, the biopesticide was maintained at 42 degrees for 3-4 days. This is to show you the dehydrin that are in structure. There is no secondary structure, no tertiary structure, but they took the structure of the protein, the interacting protein. This is the purification of layer 2. We can skip this one. Here we tested on using the LDH, lactate dehydrogenase, activity at 50 degrees, and it is clear here, layer 2 from date palm enhanced the thermostability of the enzymes, and the relative activity is somehow maintained. So here it came the idea to formulate one biopesticide using this overexpressed dehydrin. Of course, it's costly because we produce dehydrin, we purify, and then we add it to a commercial biopesticide. But this was the concept, so we have to demonstrate that. We did it, and we obtained a good results for the mortality of eggs in eggs hatching, for the larvae, and also for the adults. And then we formulated our concept, we protected ourselves with the one patent, and then we went to make a new BT. We generated new BT by making a recombinant or transgenic, if I can say, Bacillus thuringiensis tenebruensis, overexpressing the dihydrin from date palm. And this is in the process for patenting. So I have one video to show you. The discussion will be to win some time. This is the paper that we published in the patent. And here I have one video to show using AI. I hope it will work. Why is not working? I'm doing that. If the technician can help to get this video. It worked in my full screen before this one.
It's not work.
No, it dropped.
Not work. Yes. Sorry for that.
Anyhow, so this is, I can explain this. This video is 2 minutes, I think, to show how we did it. So the first step is to we overexpress the dihydrin in expression vector. Then we cloned it. We expressed in expression vector. Then we went through large-scale production, purified using histidine tag colon. Then we added to the BT, and then we tried in not in the farm, but we tried in the lab using trials with insect larvae and adults. And the new one that we did is all the first step was cut it off, and we made a new BT overexpressing by itself the dihydrin, and is heat tolerant to 45 degrees, which is, I will not say it will stop the red palm-weevil, but at least it will control the spread of the red palm-weevil from farm to farm and from date palm to others. So it will make the spray of red palm-weevil efficient for a few days, not for a few minutes. And thank you. I'm happy to ask if you have any question.
Thank you. Thank you. Thank you, Dr. Khalid. Is there any question? Somebody would like to ask a question? Please. Would you like to come over, because we just have this microphone here?
It's okay.
Okay.
Go ahead. Yes. Let's say the most important track in dehydration is the lysine track. Is the most important. So we did some mutation in dehydrin with K segment, serine segment, serine lysine segment, YS, YK, and so on. And we found that the K segment is the most important in the dehydrin. So even if you transform only a peptide containing the K segment is enough to get the protection of the other molecules. Yes, we did fusion. We did fusion. Yes, yes, yes. Yeah, we can fuse it. We didn't fuse it right now, but we can fuse it with the cry genes. Yes, for the BT. Yeah. Okay. Any other questions? There is plant, we did it in different plants. We did it in.
Can you hear me? Yeah? So the dehydrins or the layers are really known to improve heat tolerance.
Yes.
So are you
Drought and salinity.
Yeah,
Yes.
yeah. So do you have anything related to
Yes, we have some plants. We did it in Arabidopsis, and we have Arabidopsis transgenic plants that are drought tolerant and stress tolerant up to 200 millimolar sodium chloride. And we have some, because it has a pleiotropic effect. It activates many genes. Some of them is like for the oxidative stress, we have I have some jasmonate for the resistance to pathogen, and for the for the oxidative stress, the salicylic acid. So it's we did some. It's a long way. This we did it with dehydrin from wheat, and we found that the transgenic Arabidopsis plant overexpressing this dehydrin really improved the tolerance to salinity and to oxidative stress. Yes.
Any other questions, please? Okay, let me ask, is Dr. Zilal here? No. Dr. Robert Fryer? Dr. Abdul Rauf Muhammad? Dr. Muhammad Kamal Abbas? Okay. Okay, well, thank you so much. Thank you. Okay. Are there any questions or comments to raise? Please. What? Can you use the microphone?
Are you concluding the session now?
Yeah, we are concluding, yes.
Yes.
My presentation is still awaited. I am Waqas Vakil. I was supposed to deliver at 11:15.
Who is?
Waqas Vakil, yeah.
Waqas, Wakil, yeah.
Waqas, we called you, but you were not here.
I was in room A, but I am...
Yeah, but we called you, so please. Yeah, please, the floor is yours. Yeah. Yeah, please. If any of the speakers who did not speak is here, please just raise your hand. Please.
I was supposed to be here. Thank you very much. Excuse me. Can you release this presentation?
And please introduce yourself.
Thank you, everybody. Can you hear me? Okay, thank you very much. And actually, I was supposed to deliver my talk at 11:15, and I was in the room A, and then I arrived at 11. Ultimately, I waited for all the participants, so that to finish that. Again, I'm Waqas Vakil, and I'm a professor of entomology at University of Agriculture, Faisalabad. At the same time, I'm a guest scientist at Muechenberg Research Institute in Germany, in Muechenberg. My topic of today is to assess the combined and individual application of the different control agents for the integrated protection of the Rhynchophorus fragaensis. This study we have conducted in collaboration with Greece from the University of Agriculture, Athens, with Nicholas as well. You are well aware, obviously, that date palm is a global of the importance of the crop, and Pakistan is at about at fifth and or either on the sixth number in the world's date palm production, and it has a very significant impact and the role in the economies of the Gulf region as well. Rhicophorus fagansis, one of the most notorious insect pests which invaded Pakistan as well as started from from Thailand. Some says we have published this data that it invaded from the Thailand, and some data, all the literature shows that it invaded from the Indian Peninsula to the Gulf region, and ultimately now it reached to the California, to the France, to the palms, Canary palms, and everywhere throughout the globe. It's all over the world over there. It has a broad range, and obviously canary palms and all of the palms, even the ornamental palms, are also affected due to the red palm weevil in the world now. These are the these are some of the symptoms which are all were obviously well aware of these symptoms, how it damages to the trees of the palms and as well as the date palms. They are very much clear, and due to their cryptic and concealed life cycle, it is very difficult to detect at earlier stages. Obviously, there are the many other challenges like economics, environment, there are so many aesthetic losses. Chemical insecticides are not effective against them, red palm weevil, because of the concealed life cycle. Pheromone traps may attract them, but they are costly and highly not very effective in the high climatic regions, as well as the sterile insect techniques. They are limited due to the hidden mating. So all of these challenges call us for the biological control, also to protect the environment and to provide the greener approach for the controlling of the red palm weevil. But that necessitates the inclusion of the biocontrol agents for the controlling of the red palm weevil in the true real forms. We have conducted a study. This was basically a study of evaluating the entomopathogenic fungi. These are the well-known fungi, which Beauveria bassiana and Metarhizium, and they are well known due to their effectiveness against a variety of the insect pests, including the date palms, and the literature is available. But we have tried and we have tested the these entomopathogenic fungal isolates, which was isolated actually from the local region, from the Pakistan, as well as from the neighboring countries like from the India, and also from the region that are like the Bahawalpur, the Pakistani. Some of my colleagues are sitting here, the Bahawalpur, or where the temperatures are very high. We try to collect the sample, fungal isolates from that region, so that to have their more potential, to see their more potential under the high temperature conditions to control the red palm weevil. So in this effort, we have collected many of the samples. Obviously, we have isolated them and ultimately tested them at the preliminary basis to check their efficacy against this weevil in the laboratory. The basic aim of this study was obviously to evaluate their efficacy as well as to have evaluate or to have the data about the assessment on their assessment of the horizontal transmission in the most virulent isolates for this, and also to check their effectiveness on the oviposition, their fecundity, eclosion, and the larval survival and the adult survival as well. Okay, very simply and briefly the materials methods. Red palm we have collected and reared the red palm weevil from the Bahawalpur region, that is a region on the southern part of the country, and also we have reared because it has a very good sugarcane is a very good substrate to rear them in the laboratory. Red palm weevil, and we have reared and have maintained a very good colony for the last 10 years in the laboratory at University of Agriculture, Faisalabad. The temperature and humidity was maintained in the laboratory for the rearing of them. We have isolated the fungal isolates. We have collected the soil samples, the insect cadavers from the different sources from the different field sites, and ultimately we have mass reared and isolated, identified both on the molecular basis as well as onto the morphological basis, and ultimately we have the good culture maintained at the laboratory in the university. We have deposited these isolates into the plant pathology department in the fungal that is a bank, a fungal culture bank. Obviously, we have tested to see the dose response assays. We have tested about 25 isolates against red palm weevil, and the results were very promising. The results says that we have checked also the mycosis as well as the mortality, and the mortality and mycosis was very good in case of the 3 of the isolates that were the among the 25 ones that were isolates. That was the 20G WGW G2531 as well as the other 12. The other in case of the same, obviously the mycosis was also recorded, and mycosis also gave the same trend of the results. This data was for the larvae, and for the adults the same results were for the dose response assays. The same data has been exhibited for the adults. In case of isolates, 100 only WG30, 23 and 25 got successful, which gave us about a 100% control of the red palm weevil, and after the 14 days, and the mortality reached up to the 80% in case of the WG30 25. In case of the adults, with regard to the hatching, decreased with the higher concentration of the these 3 fungal isolates, and there was a varying level of the mortality of the red palm weevil in the laboratory. However, the WG25 one of the isolate, that is WG25, caused the lowest hatchability, or that is about 50% up to and that at the both concentrations which we have tested in the laboratory. WG again 25, one of the isolate that caused the highest mortality in case when we have tested the horizontal transmission ability of these fungal isolates, and horizontally they have infected male, and the female concentration showed the strongest effect when they were both of the treated, and the infected of the both of the male and the female. However, in virulence ranks, WG25 was most potential, and the WG8 isolate, which have been deposited in the microbial bank of the plant pathology department, that showed the less effectiveness against the red palm weevil. Again, this is a horizontal transmission, and the same trend has been followed by all of these. 23 and 25 was the most effective ones. This is the data showed. We have also tested EPNs in combination with the most virulent isolate of the Metarhizium that has been tested, and with this result, the results exhibited that the combination gave us a very good mortality data. However, I am showing only the 2 populations. One have been collected, from the Multan on the southern side of the country, and the Bhakkar is also in the south of the country as well. These 2 populations were tested for assessing the effectiveness of the combination of the most potential isolate of the Metarhizium, as well as in the combination with the EPN commercial formulation of nematodes, and they gave a very good pupation. Adult control of they gave us a very good control in combination treatments of the red palm weevil, and resultantly there was a less pupation. Adult emergence was very low in case of the combined treatments where it was. Similarly, the larval duration and all of these factors, larval duration, larval weight, prepupal duration, prepupal weight, prepupal duration, all have been affected according to the same trend as was observed in the case of the mortality of the red palm weevil in the laboratory. Same was true for the longevity of the adult, both of the male and the female, and the adult weight as well. Conclusions: both of the isolates which have been isolated from the southern part, they gave us a very good control even under the higher temperature regime that was about 40 Celsius, and because of being the native isolates, we have a very good results, and there were other conclusions that has been already described. the field application testing is very much important, and we are going to conduct the field experiments in the Bahawalpur, one of the commercial farm, as well as into the government level at the one of the government organization in the Multan region. That is also on the south of the country. Thank you very much for your attention, but I would like to take 2 more minutes from you for introducing, because I'm also at the same time the convener of the integrated protection of the date palm. That is basically a working group under the International Organization for the Biological Control. Myself is the convener, and Nicholas Papalidis is from the Greece. He's the liaison officer. Basically IOBC is an international organization for biological control. They have the 6 regional sections. WPRS is one of them, and they have the 26 working groups. Integrated protection of the date palms is one of the working group in the world. That has been established in 2016 in Bahrain, and the Professor Aziz was the first convener at that time, and the conference was very successful. The proceedings were published over there in the Gulf University. These are some of the glimpses. This is the basically abstract book, rather the proceeding book. There was another event that happened 4 years back at the same location, and we have organized a very good symposium over here. Recently, we have organized a conference in Algeria, in Biskra, at the Date Palm Research Center at Biskra in Algeria in November. And we have about 40 international delegates into that meeting, and that was a very successful event. The proceedings of this meeting has also been already published, which have been conducted in Algeria in November. And some of the selected papers are going to be published as a special issue in the Phytoparasitica Journal. I would like to invite all of you to join this group, Integrated Protection of the Date Palm Working Group, and the Facebook page, and also other social media. You can find this group, and you are welcome to join us as a group under the IOBC platform. Thank you very much to all.
Thank you, Dr. Ukas. Dr. Abdullah, for the online, please.
The floor is yours, Dr. Zilal. Just a second, please.
No sharing for my screen?
Yes, please. Introduce yourself first, please.
Yes. I am Zila Suleiman Al-Kadur. I am affiliated with Wageningen University and Research, and today I am, and also as a PhD candidate at my last year of PhD. And also, I am a researcher and lecturer by Iris University of Applied Science in the Netherlands. Today, I'm pleased to present my research titled Endophytic Establishment of Weveria bassiana in Date Palm. This work explores the sustainable biological strategy for protecting date palms against one of the major destructive pests, the red palm weevil. And thank you for being here, and I'm looking forward to share this finding with you. So date palm weevil, especially Phoenix dactylifera, where this study focuses on, are highly valuable in arid and semi-arid regions. So they are important for food production and for food security, but also ornamental landscaping. So, however, this important crop faces a very important and very destructive insect, as I mentioned, the red palm weevil. Red palm weevil, as most of you, all of you knows, that all its larvae and almost all life cycle stage are hidden inside the trunk. which sorry.
Excuse me, will you open the slides? Yes. One second, one second. Can you minimize the web browser window?
Would you please share your presentation? We cannot see your presentation. Would you please share your presentation? Please. It's okay.
Yeah?
Thank you. Yeah, it's okay.
Okay, thank you. So, yeah, here. Now, I will continue. So, the larva stage of, and most of the life stages of red balm weevil are hidden inside the trunk of the balm trees. So, the early detection for this insect is almost impossible. And chemical treatment, yeah, for me and for the whole world, I'm sorry, but I have to say that it's no longer acceptable. And the traditional control methods alone are no longer sufficient also. So new sustainable approaches within integrated pest management systems are urgently needed for to control or manage these insects. Within my previous studies, master studies, we tested different entomopathogenic fungi isolates against red balm weevil, and the results were very positive. So the fungi were able to kill the insect within 4 days in a high efficacy rate. Beauveria bassiana, one of those entomopathogenic insect fungi isolates, and those kind of fungi are not only able to infect and kill insects, but they are also able to localize or to live inside the plant tissues as endophytes without causing any disease for the plant. Now, the biology of these insects as hidden stages inside the trunk of the plant and the large canopy and structure of balm trees that make the application for any treatment very difficult, and the positive efficacy of Beauveria bassiana and the biology of Beauveria bassiana of entomopathogenic fungi in general as endophyte inside plants, led us to ask a question. What if plants could defend itself from the inside out? So instead of spraying fungi only on the outside of the plant, we investigated where we could establish the fungus inside the balm itself, creating an internal biological system defense. And so within this study, we focused in two questions, actually. So do white date balms naturally host beneficial endophyte fungi? As a first question. The second one was, can Beauveria bassiana be artificially introduced into date balm in practical and effective way? Together, those two questions help to determine whether endophytic pest management is a realistic strategy to manage red balm weevil. So, to answer the first question, we examined our 6 untreated field-grown balm trees has been sampled in 5 coastal areas in Syria. And from those plants, we got 200 leaf section and examine it to see if we are able to find the fungi or not inside those plant tissues. 5% approximately from the samples we examined found it infected with the fungi. And that means there are 200, there are not that much samples. So from 200 leaf session, we were able to isolate Meteorrhizium, Cordyceps, Acanthamyces, and Beauveria bassiana isolates. So this result confirms that date balm can naturally host beneficial fungi, supporting the biological relevance of our concept. I called the isolates we collected from those areas as EPA and EPH, and I really advise you to focus on these two pictures here, where we got the fungi from. The plants that we collected or sampled were wild palm trees, not treated at all. So next, we tested 4 isolates of Beauveria bassiana at concentration of 1 to the power 8, so not a very high concentration, 1 to the power 8 conidia per milliliter, using 1 to 2 years old greenhouse palms. The isolates we used, 2 isolates derived or isolated from red balm weevil during previous studies in Syria, from Syria, and the other 2 isolates, the EPH and EPR, we isolated during this study in the previous experiment. So this design allows us to compare between 2 sort of isolates from insect origin versus isolates from plant origin. We compared 2 delivery methods. The first one, leaf injection, trunk injection, we hear leaf injection. We injected 30 microliter of the fungal suspension into the leaf tissues and sealed the injection point with parafilm. And we tested the localized vascular uptake of this fungal suspension. The second delivery method was spray. We spray 30 milliliter of the fungal suspension as a fine mist on the canopy of the tree. If you can see here, this part is the treated part. And in the same tree, from the same tree, we covered one front with plastic sheet and not treated, if non-treated, and we are not able to call it as a control because in the same tree, but I called it as a covered treatment. And here, with the injection treatment, we sampled our samples from above the injection point and below the injection point, and the control samples, if I can call it like this, it's from another front in the same tree, and I call it away sample. So after this, after sampling this, so the result shows that the trunk or tissue injection created a rapid and highly localized colonization, peaked around day 15, and the highest colonization, about 80%, occurred and directly above the injection point. Below the injection point, colonization around 45%, and from the away of the distance samples, that the front showed of the colonization rate showed 35% of colonization rate. Between the 2 isolates we tested here, the isolates derived from the insect, we didn't see any significant differences, just between the control of the away, of course, and the treatment was and significant difference. So this result told us injection is effective for fast establishment, but the fungus spread remains around of concentrated near to the treatment point. Foliar spray behaved differently. Colonization developed more slowly, becoming detectable at day 10. And importantly, it persisted till day 55. For the endophyte, and that was for the insect-derived isolates. For the endophyte isolates, the EPA and EPH that we took from the plant tissue or recovered from the plant tissue, they achieved also strong colonization level, approximately 50% from as colonization rate. This demonstrates that spraying can successfully establish the fungus internally while offering a practical method for large-scale use. As I mentioned, that treatment for date palm is not easy, any kind of treatment. So an interesting finding here was the fungal origin has a little effect on the performance of this fungi inside the plants. Whether isolates came from insects, red balm insect, or from the palm tissue, the colonization successful was very similar. So that suggests that long-term persistence is influenced more by the plant physiology and the environmental factors more than the origin of the fungal strain for establishment purposes. However, one important point must be emphasized here, that the living biological protection, and I like to call it like this, living biological protection is effective but not permanent. So across all the treatment isolates, individual isolates that we use, the presence is gradually declined over the 55 days period. That, therefore, in practical agriculture, timing of application is critical. Reapplication protocols will be necessary to maintain the biological shield during peak red palm weevil farming seasons. So reapplication in these terms, and here I just designed like in concept or for integrated pest management systems for red palm weevil for applying this method within this system. So in conclusion, this study validates the first step in biological control. We demonstrate that Weevil bassiana can be practically and sustainably introduced into palm trees or into the palm tissues. But field validation, of course, we need to be done or is needed. So to determine whether this internal colonization can directly reduce red palm weevil attack. So in the lab, within the studies, we noticed that Beauveria bassiana, or let's say entomopathogen fungi, because we tested not only Beauveria, but also Metarhizium, Acanthamyces, they were able to kill the insect within 4 days in high percentage. And we were able to introduce the fungi artificially inside the plant tissue, so to make the level of those fungal spores inside the plant in a high rate, as if I can say. Now we need to see in a field application or in a practical way if we are able to determine that this establishment method is able to reduce red palm weevil attack. And this open, of course, a promising pathway toward environmentally responsible integrated pest management in date palm. So thank you very much for your attention, and I would be very happy to answer your questions. And this, my contact information is shown here for any, if you would like to discuss collaboration in the future or any other details. Thank you very much.
Okay, thank you, Dr. Zilal. Thank you so much. Is there any question? Here, somebody has a question, please. Okay, well, thank you so much, Doc. Thank you.
Welcome.
Okay. Is any of the speakers who didn't join us this morning here? Okay. Are there any questions for any of the speakers? Okay. So we will break. I think lunch is at 12:15. So we will be having a break, but we will be back here at 2 o'clock. To see if some of the people who didn't show this morning will be here, and also the people who will be in the afternoon session will be with us here. So I'll be here at 2 o'clock. So please let us convene here at 2 p.m. Thank you so much. Thank you.